protein coupled receptors, and calcium. As usual, I don't represent any medical facility or institution in these lectures, and the use of any graphics is for educational purposes only. Please do not monetize any of this. So a signal molecule is going to bind to the G protein coupled receptor, and that's going to activate this G protein. So as you might remember from last time, you have an alpha subunit and a beta gamma subunit. So we're going to focus on the beta gamma subunit. And what it's going to activate is phospholipase C. And phospholipase C is going to cleave PIP2, PIP2, into IP3, inositol trisphosphate, and diacylglycerol, DAG. All right? So what's going to happen is the IP3 is going to go to the endoplasmic reticulum where there's IP3 gated channels. So the binding of IP3 causes these channels to open and calcium to enter the cytosol. What that's going to do is this protein kinase C is usually just hanging out over here in the cytosol, minding his own business. But once the calcium starts to come up, this protein kinase C is going to be recruited to the plasma membrane. And diacylglycerol, and what's not drawn here is phosphatidylserine, but diacylglycerol and phosphatidylserine in combination with this increase in calcium are what's going to activate the protein kinase C. So initially, IP3 binds here, calcium floods, this protein gets recruited to the plasma membrane, diacylglycerol and the calcium and phosphatidylserine work together to activate protein kinase C. And then protein kinase C is going to phosphorylate its targets, and that's how you're going to get your signal cascade. So acetylcholine, which has receptors on certain endothelial cells and certain blood vessels, is going to bind to its G protein coupled receptor, its GQ receptor, that's going to activate phospholipase C, and that's going to lead to inositol trisphosphate, that calcium cascade, and eventually the activation of nitric oxide synthase. This is important. You must know this. Nitric oxide synthase is made in the endothelial cell by combining arginine, oxygen, and citrulline, and then it diffuses into the smooth muscle. All right? The nitric oxide made in the endothelium as a result of phospholipase C and GQ protein activity will diffuse, this nitric oxide will diffuse into the smooth muscle. Once it's in the smooth muscle, it's going to act on the nitric oxide receptor, which activates guanylyl cyclase, which isn't drawn here. But the main point is that that's going to increase levels of cyclic GMP, and that's going to activate protein kinase G, and that is what relaxes the smooth muscle. All right? So let's say, just for argument's sake, that you're a heavy chain smoker, right? And there's other mechanisms that make nitric oxide synthase, not just this pathway. All right, but let's say you're a heavy chain smoker and that's been established to destroy the endothelium. Now your source of nitric oxide synthase is knocked out. You're not getting nitric oxide from the endothelium anymore because it's damaged. What do you think is going to happen to the smooth muscle? It's going to contract. You're going to enter a state of vasoconstriction. All right, and this state of vasoconstriction is what we call bad because that's going to cause hypertension. That's going to eventually cause end organ damage. So this is just one of the many ways in which smoking kills you, is that it destroys your endothelium, and that damage to the endothelium is what prevents smooth muscles from relaxing. All right, not why? Because you no longer have nitric oxide. All right. Now, when we look on the right, I just want to point out that the healthy functioning of platelets depends on a lot of different pathways, not just GQ pathways. Don't worry about this pathway on the left, but it's a unique. It's not G stimulatory. It's not GQ. It's not GI. It's its own thing. It's a Row-Jeff pathway that you don't really need to worry about. But notice how there's two different pathways, GQ and GI. Right? In the last video, I talked about GS and GI. Notice how multiple signaling pathways can converge on the same result. All right? Multiple signaling pathways can converge on the same result. That's the importance of having different pathways and different levers and different mechanisms so that a cell can respond to different stimuli in different contexts. But what I want to point out is that the ADP, how ADP can bind to GQ and GI. All right? There are also unique ADP these platelet receptors, by the way. The P2Y12, especially the P2Y12 receptor, can be a pharmacological target. All right? And there's a very important medication that you need to know, and its name is clopidogrel. And while it's not really involved in the GQ, oops, that's a misspelling, that should be an O. And while it doesn't inhibit P2Y1, it does inhibit P2Y12 activity. In fact, there's a common method of treatment called antiplatelet therapy where you give aspirin. It's called dual antiplatelet therapy. That's very important in the treatment of vascular accident. What they're called vascular accidents, when platelets have clogged up a vessel, and part of the treatment of that and the prevention of future events is called dual antiplatelet therapy. And the clopidogrel is going to bind the P2Y12 receptor. So the clopidogrel, I might be pronouncing it wrong. The point is, this is going to act on P2Y12, and that's very important. And that prevents, and don't memorize this right now, I'm just planting the seed for later, and that inhibits the G2-3 glycoprotein complex. Or I'm not going to even write it out here because I don't want you to try to memorize it. Just know that it inhibits this interaction. All right. The other thing I want you to know is that there's a thrombin. This part of the pathway is inhibited. And what is it inhibited by? By aspirin. All right. So both of these are inhibited by aspirin. So aspirin covers this, and clopidogrel covers this. All right? That's important. You must know that. That's dual antiplatelet therapy, because what happens at the end of dual antiplatelet therapy, you have GQ blockade and you have GI blockade. All right? You have GQ protein inhibition and GI protein inhibition. All right? So these are, I promise you, you will be tested on these at some point. I promise you, you will be tested on this slide. All right, you absolutely will be tested on this at least once, because we talked about phospholipases in the context of GQ protein coupled receptors, but I want to talk about it more broadly now. Okay, so we said that phospholipase C is going to make diacylglycerol and IP3. Diacylglycerol is going to go into the arachidonic acid pathway. All right, and other phospholipases will also make diacylglycerol to enhance the arachidonic acid pathway. Namely, and one you might see on exams, is phospholipase A2. That's very important for inflammation. So how do we stop inflammation? We can just stop it before it begins at the level of phospholipase, and we do that with steroids. All right, but the problem is steroids are you can see how upstream, so how high up they're acting. So there's a lot of side effects. You know, the higher up something is acting, the more side effects there are. So we can go down a step and focus on cyclooxygenases, which eventually lead to the formation of prostaglandins, and these mediate a lot of inflammation. All right. So what's going to inhibit those? Your NSAIDs, Tylenol. So NSAIDs include things like ibuprofen. Tylenol would be acetaminophen. Aspirin can inhibit cyclooxygenase. And then you have selective COX-2 inhibitors. All right, selective COX-2 inhibitors. And the reason for that is COX-1 is very important at baseline to maintain the integrity of the mucous membranes. And that's especially important in the stomach, where there's so much acid and so much damage that can be done by that acid, that you need to have this protective mucus so the stomach doesn't destroy itself. And that's what's important to that is cyclooxygenase 1. And NSAIDs are non-selective. They are non-selective. So they block COX-1 and COX-2. So what you can do is you can use a selective COX-2 inhibitor, which spares COX-1 function. All right, so you knock out COX-2, which is associated with a lot of the inflammatory pathways, and it leaves COX-1 alone, because COX-1 is expressed in all tissues, in all mucosa, in all tissues, in a lot of different tissues, at some baseline level of function. That's important. So you can use a selective COX-2 inhibitor, especially if you're concerned about protecting the gastric mucosa. All right, and then you also just have your Tylenol, and aspirin. All right, and then on the other arm, you have the lipooxygenase pathways. I'm not going to go too much into detail for these. This is more important for asthma. A lot of the asthma pathways are involved um, with inhibiting either the lipooxygenase pathway directly, and that's going to be a medication like Xyloton, where it just inhibits this, or you're going to inhibit where the leukotrienes act. All right, and that's going to be very important for asthma treatment. Um, so those would be like the Lucast medications, Montelukast, Zaferlukast. The Lucast, when you see Lucast, it's antagonizing the leukotriene receptor. All right. So calcium is such an effective signaling mediator because it's present at baseline in low concentrations within the cell. So once calcium levels do go up in the cell, it's a huge signal that something is going to be a very fast acting, rapid response, positive feedback loop. And that's because it's so low concentration at baseline that even small fluctuations can trigger a response. So that's why it plays such a central role in different signaling pathways and hormone pathways, muscle contraction, synaptic neurotransmission, um, and in turn, that is why hypo and hypercalcemia can cause so much dysfunction. That's why conditions like hyperparathyroidism, which causes too much calcium, and hypoparathyroidism, which means not enough calcium, um, that's why they can affect so many organ systems. All right, because that disruption of calcium balance is disrupting a lot of signaling. So there's a mechanism in which stimuli such as depolarization, stretch, etc. will activate calcium channels in the plasma membrane. And that will activate so then calcium is going to flood into the cell. And remember, in the endoplasmic reticulum, there's a lot of calcium channels. One of them is IP3 gated. One of them is the ryanodine receptor. And calcium that enters the cell through the plasma membrane will bind to the ryanodine receptor. So it might be better if I draw this. So let's say calcium has entered the plasma membrane, and this is the endoplasmic reticulum, and there's a calcium channel here gated by a ryanodine receptor. The calcium that came in from that earlier signal, whether it was depolarization, stretch, whatever it was, whatever the signal was, and it caused calcium to come through the plasma membrane, that calcium is going to bind to the ryanodine receptor. Okay, calcium is going to bind to the ryanodine receptor, and that's going to cause calcium to be released from the endoplasmic reticulum. And that is called calcium-induced calcium release. All right? So, stimuli can activate plasma membrane calcium channels. That's going to cause calcium to come in and activate ryanodine receptor-gated channels in the endoplasmic reticulum, and you're going to get calcium-induced calcium release, which is you can imagine that causes the all or nothing response for calcium. So let's say you're in a nerve, the result of this will be all this calcium will lead to exocytosis of synaptic vesicles. So like in the nerve, you have all your neurotransmitters, and they're hanging out in the synaptic vesicles, and then they have to be released, right? Here's the dawn. So when calcium-induced calcium release occurs, these vesicles will fuse with the synaptic membrane and release the neurotransmitter into the synapse. I'm not going to go into detail here. I'm just using it to illustrate how calcium-induced calcium release is important. All right, and there are other signals that independently release calcium from the ER, like GQ proteins. Right, so that's one of the types we just talked about. All right. So I hope this wasn't too bad. It's a lot of information, but it's a lot of important information. Good luck.